The following is a project supported through the Canadian Agri-Safety Applied Science Program, administered by Agrivita Canada. Fugitive emissions following manure spreading, risk assessment, and engineering controls. Manure spreading on agricultural land is a common practice in many parts of the country and helps benefit crop production. Aside from the obvious side effect of unpleasant odors, manure spreading can contribute to pollution and pose a potential hazard to human and animal health. Human and animal pathogens, harmful compounds, and antibiotic-resistant bacteria are present in livestock wastes and may be aerosolized in large quantities when spreading on agricultural land. Once airborne, these contaminants can then affect the health of workers, animals, and surrounding rural communities. There is currently a lack of knowledge regarding airborne contaminants associated with manure spreading. This project will quantify emissions of airborne contaminants to provide a better understanding of the health risks associated with manure spreading. Work will also be done to determine the best strategies to mitigate these risks. In order to quantify emissions from manure spreading, the research team built a custom large-scale wind tunnel to conduct experiments in a controlled environment. The tunnel is made from a large greenhouse and is equipped with different features: 10 fans at one end to create a controlled airflow rate, a large wooden box filled with soil to replicate a piece of land, and a watering system to simulate rain. Environmental parameters including wind speed, temperature, and relative humidity are monitored continuously in the wind tunnel. Samples are collected before, during, and after the manure spreading experiments to quantify gases, odor, bioaerosols, and dust. Experiments were conducted with solid cow and poultry manure with a small horizontal beater spreader. Two types of spreaders were used for liquid swine manure, a splash plate system, and a dribble bar spreader. For all manure types, carbon dioxide and methane emissions occurred mostly during the first 10 minutes from the start of spreading and then stabilized. Ammonia was released more progressively, and emissions were highly variable among the different manure types and spreading equipment. Ammonia emissions were actually more correlated with the air temperature. For all the spreading tests, the odor intensity was highest during spreading and then decreased to near baseline values after 30 minutes. Odor intensity values were highest during the spreading of liquid swine manure with the splash plate. For total bacteria, high emission rates were observed during the spreading tests. When comparing the different types of manure, emission rates were highest for poultry manure, followed by cow manure and liquid swine manure. With the emissions properly quantified in a controlled environment, the second objective of this project was to evaluate the health risks for workers, animals, and rural communities. An intensive field sampling campaign is currently being carried out to measure airborne contaminants and evaluate risks under actual field conditions. A series of preliminary spreading tests was performed using liquid swine manure with a splash plate spreader. The sampling methodology was adapted and used to measure concentrations of contaminants at 6 locations in the field, including a mobile sampling point behind the spreader. Similar to the tests in the wind tunnel, methane and ammonia concentrations increased following slurry spreading in the field and then stabilized over time. For odor intensity, the maximum value measured was only half of the maximum value obtained in the wind tunnel, illustrating a higher dilution in the open field. The total bacteria concentrations decreased as the sampling point was further away from the spreading zone. In addition, several antibiotic-resistant genes were detected in the liquid swine manure used for these tests. The air samples are currently being analyzed to determine if these antibiotic-resistant genes were aerosolized during spreading. Additional field tests will be carried out during the final year of the project with other types of manure using different spreading techniques. A promising technology that can inject solid manure directly in the soil will also be tested to reduce airborne emissions. Results from this study will help identify critical factors that could influence gas and bioaerosol emissions during manure spreading. It will also be possible to evaluate the health risks associated with manure spreading and to recommend mitigation strategies to protect the health of farm workers, livestock, and rural communities. Please visit our website for more information on all the projects of the Canadian Agri-Safety Applied Science Program.